literature, you can actually find at least once a month a new type of cancer in which an ALK fusion has been detected by next-generation sequencing technologies. ALK is also altered by amplification, particularly in neuroblastoma. Point mutations that activate the kinase domain outside of ALK fusions in neuroblastoma has also been reported in anaplastic thyroid cancer. And then most recently, this ALK ATI variant has been reported. So ATI stands for Alternative Translation Initiation Site, was found in 9% of melanoma samples in one study that was recently reported by groups at Memorial Sloan Kettering. So another point I want to say before we really get into thinking about the next generation therapies is that there is actually not just one ALK fusion. There are numerous ALK fusions. We spend a lot of time talking about the different mutations in EGFR which confer sensitivity or resistance to EGFR TKI therapy. But just to point out that we actually don't really think right now about ALK fusions and how they're different, but there are several different fusions that have been described in lung cancer and numerous fusions in the other cancers as well. The most common fusion variant that we find in lung cancer is a fusion of EML4 to ALK, but even within EML4, there are multiple different breakpoints within the EML4 gene that fuse to ALK, causing numerous different variants, and I will discuss that a little bit later on in my talk. So this is the phase 1 trial of crizotinib, which has really set the stage for ALK TKI therapy in patients with advanced lung cancer. So just to go back to the past, this study had patients who were heavily pretreated. All were found to have ALK fusions by using ALK FISH, and the response rate in this trial was 60%. So why am I showing this? So this is crizotinib was FDA approved now more than 4 years ago, and though now we have several different ALK inhibitors that are FDA approved, most recently alectinib yesterday, and I'll show you some of that data. So the question really becomes, and the focus of my talk will be, what do we do when crizotinib fails? How do we treat patients after they develop acquired resistance to crizotinib, and what does acquired resistance to crizotinib even look like on a molecular level? So we start to think about molecular mechanisms of acquired resistance to crizotinib, and keep in our mind what acquired resistance to EGFR TKI therapy looks like. The first notable thing is that first, ALK resistance, or crizotinib resistance specifically, is mediated by second site mutations in the kinase domain that actually fall over the entire kinase domain. So unlike T790M in EGFR, there are numerous second site mutations in the ALK kinase domain that occur at various portions of the kinase domain and confer varying levels of sensitivity or resistance to the different ALK second generation TKI therapies that we have available in our armamentarium. This is a review that came out this year looking at the frequency of some of these second site mutations. And just to point out, we actually don't even have a great estimate of the frequency of these mutations as they occur with crizotinib resistance. But one important analogy to make is that there is the gatekeeper mutation. It's L1196M that is analogous to EGFR T790M. And one last point on this slide is, unlike T790M, which is found in 50 to 60% of patients with EGFR TKI resistance, these second site mutations, and there are numerous second site mutations, actually occur in the minority of patients with crizotinib resistance. So there are numerous other mechanisms at play here that are being elucidated using preclinical and clinical studies. So next generation ALK inhibitors is actually a lecture that could take a couple hours to give because there are so many next generation ALK inhibitors. So I tried to simplify and make tables that would facilitate discussion and also be useful for reference later. So how do we think about these second generation ALK inhibitors? Well, first, on the far left-hand corner is the different ALK TKIs. Most of them have names now. The second is the company that is producing these or marketing these different ALK inhibitors. The third and an important column is what are the other targets of these ALK inhibitors, and they differ. And just to point out a few, so several of these second generation ALK inhibitors are also ROS1 inhibitors, analogous to crizotinib. So brigatinib is a ROS1 inhibitor, lorlatinib is a ROS1 inhibitor, and then entrectinib is a ROS1 inhibitor. Notably, ceritinib does not have off-target ROS1 activity, or the second most common targets of ceritinib actually are insulin-like growth factor receptor 1 and insulin receptor. And then finally, what is the status of these agents is another way that we can differentiate them. So like I mentioned, alectinib was just FDA approved yesterday for the treatment of patients with ALK-positive metastatic lung cancer who have progressed on or become intolerant of crizotinib. Likewise, ceritinib was approved last year for the same cohort of patients. So just to review the ceritinib data, this was published last year in the New England Journal of Medicine. This is a waterfall plot of the ceritinib trial with an overall response rate of 56% of patients who who had previously received crizotinib and 58% in patients who were crizotinib naive. If we look at that subset of patients with crizotinib resistance, there is only select patient samples that were able to be studied on a molecular basis to actually understand what was driving resistance in these cases. And you can see here that on this waterfall plot, NM stands for no mutation, and then there's some second site mutations, and AMP stands for amplification. So the take-home message of this slide is that though the response rate in ceritinib and the hypothesis going into the trial was that ceritinib would be able to overcome the ALK second site mutations that confer crizotinib resistance, there was actually patients both with those second site mutations and patients without those second site mutations who responded to ceritinib therapy. So there was patients who did not have a kinase domain mutation who their tumors still retained some ALK dependency. An important characteristic of all of these second-generation ALK inhibitors is that they have a CNS efficacy. So here is some of the data on CNS responses to ceritinib. This is data that was presented at ESMO last year. The overall response rate in the CNS was 54% in this data set of 246 patients with ALK-positive lung cancer, 124 of them who had brain metastasis at the time of study entry. So if we switch gears now and talk about alectinib, the drug that was just FDA approved. This is the phase 1 trial of alectinib by our friend Shreesh, who's in the audience. This was 47 patients with ALK-positive lung cancer. The overall response rate in this study was 55% with a CNS response rate of 52%. This data was just updated in the IASLC meeting in Denver in September this year. So this is, again, the study designed for the alectinib. This is patients who had progressed on prior crizotinib, who were all received alectinib. And the data is as follows. So this is data presented by Alice Shaw, again, in Denver just a few months ago. So the overall response rate for alectinib in this study was 52%, with a disease control rate of 79% and a median duration of response of 6.7%, all in patients who had received prior crizotinib therapy. The median progression-free survival was 8.1 months, and the 12-month overall survival was approximated at 71%. Again, alectinib, like ceritinib, has documented efficacy in the CNS. For sake of time, I'm not going to go into this, but just to say that pretty dramatic responses in the CNS seen with alectinib, similar to ceritinib. How about the other ALK TKIs which are being developed? So ceritinib and alectinib are clearly now both the FDA approved, are furthest along in the developmental pipeline. This is the data that was presented on brigantinib at the IASLC meeting in September. I'm very sorry that my table looks so fuzzy, but I've summarized at the bottom. So this was an overall response rate of 71%, so 50 out of 70 patients who had received prior crizotinib responded to brigantinib. 8 out of 8 patients who had not received crizotinib who were on the study actually all responded, so 100% response rate in crizotinib naive patients in the small segment of the trial. The median progression-free survival in patients who had received prior crizotinib was 13.4 months, so quite a long PFS. And then there was responses in the CNS as well. How about lorlatinib? So this is the newest kid on the block. Lorlatinib is the new crizotinib, which is being marketed by Pfizer. It is being marketed for both ALK and ROS1 rearranged lung cancer. This is the initial data and the first data that I've seen for lorlatinib that was presented at the IASLC meeting by Dr. Bauer. They had 43 patients who were enrolled on this study with an overall response rate of 47%. So that was my whirlwind tour through 4 of the ALK inhibitors that were being developed. Like I said, we could spend several hours just talking about each of these individual agents and the different data that's been presented among them. But really, I wanted to spend the time thinking about how can we actually differentiate between these second-generation ALK inhibitors now that there's going to be so many of them in this relatively small space of patients who have ALK fusion-positive lung cancer. So a couple of thoughts on how we can differentiate them. First, what are their off-targets effects? So what are the other kinases that these particular second-gen ALK TKIs are targeting? Second, what's the timeline of development? Clearly, some of them are already FDA approved, while some of them are just entering phase 1 trials. Third, what are the adverse effect profiles, and how might that affect our management and our use of these different agents moving forward? And fourth, what is the efficacy against different ALK kinase domain mutations associated with ALK inhibitor resistance, specifically crizotinib resistance? I think this is a very important point that not all the second-generation ALK inhibitors are created equally with respect to their efficacy against the ALK kinase domain mutations that have been associated with crizotinib resistance, and I'll show you some of that data. So here, again, trying to make some tables to be able to synthesize and digest a lot of information. This is a summary table of not all, but most of the ALK inhibitors that I showed on my first table, with both the common adverse events and then the grade 3 or 4 adverse events occurring in at least 5% of patients. Just to point out, so we've seen data about brigatinib causing pneumonitis that was largely resolved, it appears, with the new dosing patterns for this drug. Ceritinib tends to cause GI toxicity. And lorlatinib, which I've never personally used in patients, but what I understand from the data that was presented in the phase 1 trial was that it causes significant hypercholesterolemia, as well as some what appears to be mood disorder, so described as mild to moderate speech, memory, and mood effects. How else can we differentiate these second-generation ALK inhibitors? So going back to the point of there are numerous kinase domain mutations which can occur and confer crizotinib resistance. What are the efficacy of these different second-gen ALK TKIs against these different crizotinib resistance mutations? So I'm not showing you all the crizotinib resistance mutations here. For sake of time, just wanted to point out a couple of key ones. First, L1196M is the analogous gatekeeper mutation. It's the analogous to T790M. All of these drugs have efficacy against L1196M. There is a mutation G1202R that neither alectinib, brigatinib, or ceritinib have efficacy against. The only ALK TKI that we know so far, at least that I'm aware of, that has efficacy against G1202R is lorlatinib. So if you see a patient with G1202R as a mechanism of crizotinib resistance, lorlatinib appears to be the drug of choice for that patient right now. And then another interesting example is alectinib resistance. There's a couple of case reports on alectinib resistance being driven by V1180L or I1171T mutations. Those two mutations confer resistance to alectinib and crizotinib, but not to ceritinib. So a patient who had received crizotinib, then alectinib, and then responded to third-line ceritinib. So I think this data will become increasingly complex as we move forward, trying to understand the sequence of ALK inhibitors. So how should we sequence therapy using ALK second-generation inhibitors? Really, there's not a lot of data on this so far, mostly preclinical data. There was an interesting abstract published or presented at World Lung this year where the authors looked at what is the effects or the effects on survival if you either sequence crizotinib with a second-gen ALK inhibitor, or you re-challenge the patient with crizotinib, or you give the patient chemotherapy after crizotinib. And from the retrospective data they presented here, the median overall survival, if you take crizotinib and then sequence it with a second-gen inhibitor, of which there was many, the overall survival that they reported was 34.7 months, compared to 19.6 months with the crizotinib re-challenge, and then finally compared to 15.3 months with crizotinib followed by chemotherapy. My time's almost up. I just have a couple more slides. There is an ALK master protocol that's being planned. Several people in the room are involved with that. Stay tuned as that continues, and hopefully we'll move forward. So just a couple other issues in the last 2 minutes when we really think about next generation sequencing or next generation therapeutic targeting of ALK. What are the other issues we need to think about? First, what are the other crizotinib resistance mechanisms? So these kinase domain mutations are only occurring in less than a third of patients. What else is driving crizotinib resistance? Do therapeutic responses or prognosis differ based on which ALK fusion is present? Third, what about other histological subtypes harboring ALK rearrangements? And finally, what are the additional methods to detect and monitor ALK fusion? All of these issues really go into in my mind, think about next generation targeting of ALK. So just a couple more slides, promise almost done. These are other mechanisms of crizotinib resistance which have been described. Bypass signaling mediated by EGFR, IGFR, and c-KIT have been described, as well as KRAS and MEK mutations, and increased SRC signaling. So potential opportunities for combination therapy here. Second, if we think about the ALK fusion subtypes, I'm always surprised by how much we talk about the different mutations in EGFR, but completely gloss over the different fusions that occur in ALK. And so there are multiple different fusions, again, characterized by how much of EML4 is present within the fusion. There was an interesting study published a couple months ago looking at the frequency of these different ALK fusion variants in 74 patients with ALK-positive lung cancer. The variant 1 fusion, which is a fusion of exon 13 of EML4 to exon 20 of ALK, is the most common, followed by variant 3, which is an EML4 exon 6 fused to ALK exon 20. Does that matter? So in this paper, and I apologize if you can't see this well, this is a retrospective study where they asked the question, does the fusion, particular fusion that's present actually matter with respect to relapse-free survival and overall survival? And what they consistently found in these studies was that the variant 3 fusion, patients with variant 3 fusion appeared to do worse than patients with variant 1 or variant 2 fusions. And those are the 3 most common ALK fusions that we find, at least in retrospective studies. They also did a univariate and multivariate analysis and showed that both male gender and variant 3 fusion were associated with the worst outcomes. There actually is preclinical data. I'm going to gloss over this for sake of time, but just to show you, in particular, this is a paper on the bottom left here that was published in Clinical Cancer Research in 2012, where the authors looked at the efficacy of crizotinib against these 3 different ALK fusions and consistently found that the variant 3 fusion was the least sensitive to ALK TKI therapy. They also looked at gantenerumab, an HSP90 inhibitor, and showed that variant 3 was not sensitive to HSP90 inhibition at all, at least in these preclinical studies. So next generation histology, almost done. For patients with ALK-positive lung cancer, just wanted to point out that there are several case reports now of patients with ALK rearranged squamous lung cancer. Certainly not a frequent finding, but this is one of those case reports. They actually show the pathology in the case report, show that it is a keratinizing tumor. This patient had a dramatic response to first-line crizotinib, and the reference is on the bottom right. And just to point out that if you read the first paper closely where they actually described ALK fusions in lung cancer, this 2007 Nature paper, the first patient that they found an ALK fusion in was actually a patient with squamous lung carcinoma, not adenocarcinoma. And then finally, next generation diagnostics for ALK, or what I call beyond ALK FISH, and certainly we have Dr. Hirsch in the audience who's a big expert on this, but just things to look for as we're also thinking about sequencing our therapies. What are the other issues that we're thinking about with ALK? One of them is being detection, and some of the things that are being discussed are first, new antibodies for immunohistochemistry, new ways to detect ALK fusions using NGS on tumor samples or NGS on plasma DNA, and then finally detection of ALK fusions in tumor exosomes. There was a very interesting paper in Oncotarget a few months ago about actually using platelets to find ALK fusions, since platelets are sticky and take up the exosomes. So conclusions, future directions, and ongoing questions. First, there's multiple second-generation ALK inhibitors being developed with high response rates across the board in patients with crizotinib-resistant tumors. The optimal sequence of these ALK inhibitors is currently unknown. I think it will largely be dictated by what drugs are available, what those specific adverse events associated with each drug are, and what each drug, the efficacy of each of those drugs against specific kinase domain mutations, which can mediate crizotinib resistance. Third, will crizotinib be replaced as the first ALK TKI of choice for patients with metastatic disease? We await studies of first-line alectinib versus crizotinib. Second, will crizotinib be effective in the adjuvant setting? We talked about those trials yesterday. Fifth, will immune therapy enter into the treatment of ALK-positive lung cancer? I felt like obliged to mention immune therapy in my talk since it's so important, and there are studies, of course, we saw the data about never smokers with less response to immune therapy yesterday as well. And then finally, does the ALK fusion variant matter either prognostically or therapeutically? So this is my last slide. I wanted to say thank you all so much. I'm sorry I went over my time, and happy holidays to everyone.
Just a really quick, fantastic talk. Do you think the next generations will supplant crizotinib, or will it be the CML where, even though they're better drugs, that really the imatinib stays a primary and they become the salvage in the majority of patients?
Yeah, so I think my bias right now is that the sequencing therapy with crizotinib first, followed by a second gen. Now that we have data and case reports that patients can respond to 3 or 4 lines of ALK inhibitor therapy, I'm a little bit nervous about, can we extend the progression-free survival with a first-line, second-gen ALK TKI long enough as we see for crizotinib followed by a second-gen ALK TKI?
Some of us have moved to second-generation drugs for CML, by the way, it's first-line treatment, but there are situations where you would use second-generation drugs first, i.e. someone who presents with a CNS events. I recently had a patient like that, and I tried to get ceritinib approved and could not get it approved. I had 2 small asymptomatic events as well as bulky disease of the chest. So in a patient like that, if you would get a second-generation drug approved, what's the penetration of
Yeah, so there's actually a large, so the question is, what's the CNS response rate of crizotinib? And there's several papers on that, conflicting data about crizotinib not being able to reach CNS levels that may be efficacious against blocking ALK. So that was a big data set. And then most recently, there was a JCO paper, the first author was Dan Costa, that said that crizotinib actually has a relatively decent CNS response rate. Personally, I usually start with crizotinib just because I believe the data in the sequencing, but I think we need some more controlled studies to really ask that question, what's the best drug to start with respect to CNS management? Dr. Hirsch.
Christine, congratulations with a very elegant review. I agree with you entirely around the question about what is there a difference between the different fusion types. I'm actually surprised that we, after so many years, had ALK inhibitors. We haven't addressed that question. We have been asking that question for years. The other question I have is, we have done the fish assay for many years now, and we have quite large number of patients. Have we figured out exactly what it means to have 100% positive cells versus 80% versus 40% versus 15%, which is the minimum requirement for being positive?
Well, Dr. Hirsch, I was hoping that you would figure out that question. But I think that is an interesting one. I've seen some data from your university where patients can have very low percentage of ALK-FIS positivity and still respond to ALK TKI therapy. So I don't have a great answer to that question. I'm not sure how we would best figure that out. I think there's a lot of confounding factors with respect to just the handling of the tumor itself and the particular sections that are being used for the FISH analysis. Hopefully we'll be able to move away from that a little bit with the use of second-gen IHC and NGS.
Thank you, Kristen, for that excellent overview of ALK. I will introduce myself as Taufik Owonikoko from Emory University. And I've been charged with the simple question of, is immunotherapy the next step in small cell lung cancer?